All right. So for this class, today we're going to talk about uh, immunization, right, and herd immunity. So finally, we get into this topic that we've been hearing about. So I'm just going to turn. That should be fine. All right. So immunization, right? You're familiar with this word, right, or this term. We've heard it many, many times during COVID. It's basically the the process, right, or the act by protecting someone, right, from a particular disease, right? Usually through the use of a vaccine. Okay, and it also entails, right? The whole process, how you develop the resistance to a particular pathogen or a particular disease in this case, right? And there's several forms of immunization, and whenever you get this immunization, the state of which you're in is referred to as immunity, right? You're immune to something means that you have a certain resistance to it. Okay. And there's two forms. The book doesn't really go into a lot of detail with active immunity and passive immunity. So I'm going to talk a little bit about some concepts that is not in the book, right? Uh, but you either have active immunity or passive immunity, right? And immunization via vaccines can give you active immunity basically by producing antibodies, okay? I'm going to talk more in detail about that. So, when you get infected, your body responds to an infection, and your cells kind of assimilate that pathogen, and they start noting particular structures of the pathogen, right? By analyzing the pathogen, you can create proteins that will fight against that pathogen, right? And that's what antibodies are. And we're going to see a little bit later a little photo there. Okay, so antibodies are formed when a susceptible host during, right, its response. Um, sorry, antibodies are formed whenever you have that infection, okay, or that disease, and then that in turn promotes the formation of what are called memory cells. Okay, more specifically, they're called memory B cells. Okay, and I'll get into that in a little bit. This is what I'm mentioning that the book doesn't go into a lot of detail there. And then you can have um, uh, immunity, right? So, in essence, right, antibodies, they produce memory cells, and these memory cells essentially keep a registrar of all your infections, all the infections that you've had before. That way, it can produce a response to these infections later on if you were to get infected by the same thing again during your lifetime. Okay. So there are some other types of vaccines. They're called serum type vaccines, and they can be used. You can actually transfer a certain amount of immunity from yourself or from some host to another person, but it is temporary, right? Let's say if I were to collect a person that got infected with COVID, they produce their antibodies. You can collect their blood, you centrifuge their blood and sort out the red blood cells from the liquid and the water, right? Everything that's in the water, the ions, the proteins, is referred to as serum. So you can actually extract that and inject that into a compatible person, and they temporarily have a sort of immunity. Okay, but of course that is passive because the person themselves isn't producing the antibodies continuously, right? So that's referred to as passive immunity. If you're getting one way or another some form of proteins from the outside. Okay. So there's several ways that you can get this type of immunity, right? You can get immunity by actually getting infected by something and then just going through the infection and going through the disease, right? And building resistance, or you can promote resistance by using a vaccination, active immunity, but still you're stimulating your own immune response. So actually, that's the key point of active immunity. You're stimulating your own immune system response. Okay, passive immunity. we can see in these pictures here, could also be natural or artificial. Natural is essentially all the antibodies that basically we get from our mothers, right, are passed down to the children. You get a certain degree of antibodies, but even that would eventually wear out, right? Because the baby would need to produce their own antibodies as they get older and older and older and more exposed to the environment, right? And again, like I was mentioning, you could also get artificially passive immunity by basically a blood transfusion, for example. Okay, passive immunity there. So, uh, what are antibodies more specifically? So, antibodies are proteins. They're a class of proteins that are produced by white blood cells. So, this is why when we say like you have an infection, you have a higher white blood cell count because, of course, you're infected, you need more antibodies, so you need more white blood cells to produce more antibodies. Okay? And what happens is that these white blood cells produce these antibodies and they basically attack and neutralize the pathogens, making them harmless. Okay? They're also known, maybe some of you have heard of this if you've done a blood test, right? um, they're also known as immunoglobulins. And we have several classes of immunoglobulins, A, B, C, G, right? So there's several types of antibodies. Okay. And more specifically, again, like I was mentioning, the types of cells that produce these antibodies are referred to as B cells. Okay. So having this said, it kind of sets the stage, right? Antibodies, immunoglobulins, an infection. So that whole concept of a vaccination. So this is what you really need to know. Okay. A vaccination, what is it? So it's basically um, the concept vaccination is just basically the process that you're administrating, you're giving a vaccine to an individual, right? Via the blood, usually um, intravenous, right? Via the blood or intramuscular, uh, one way or the other, you're putting it inside a patient, okay? To with the purpose of kind of jump starting active immunity, right? Usually it's by injection. So what does it do? What does a vaccine do? It basically stimulates your immune system, right? So that it could read the pathogen, detect the pathogen, and produce those antibodies for the pathogen, right? But a vaccine doesn't have the full virulent version of a pathogen, like a virus. Generally, vaccines have a weakened, right, a deteriorated version of a virus, okay? Enough so it can stimulate your immune system, okay? What exactly is stimulating from the antibody, from the pathogen, is something called a antigen. Okay, so you do need to know these two words, antibody and antigen. And these photos here are not in your book, so make sure you understand um, what's going on here, right? So um, the vaccines help develop immunity, right, by responding to a part of the pathogen. That part of the pathogen that actually creates an immune response is called an antigen. All right. So for short, if you take a look here, anti and gen, right? Anti meaning antibody, gen meaning, what do you guys think it means? It's the same thing as mutagen, carcinogen. What would it mean? Don't you? What is it? What is a mutagen, guys? You guys remember? What would be a mutagen, Ryotaro? A mutated gene? Sounds good? No. What is a mutagen, Haruki? This is unit 3, guys. This is gonna be in the test. The gene? Kanata? Let's say mutagen. Lena? Generates. It's anything that generates a mutation. So in this case, you have anything that generates a antibody. That's all an antigen is, right? Anything that produces or makes the body produce an antibody. So you have that relationship, antigen, antibody, okay? Antigen, antibody. Where do you get the antigen? It's part of the actual pathogen, okay? It's part of the actual pathogen, all right? And what does it do? It basically triggers the response in your body, okay? So again, like I was mentioning, the pathogen could be dead, right? Scientists could just get COVID and have it broken down into a serum, inside a liquid, shake it up, and inject it in your body, take out the DNA so it won't replicate, and that's enough to essentially get a vaccine, right? In a nutshell, right? Or it could be attenuated. Attenuated means weakened, okay? Attenuated means weakened, right? Where they're slightly active just to also jumpstart your immune system, okay? So that's why when some people get vaccines, you tend to get minor symptoms because you're suddenly aggravating your immune system in your body. Your body's immediately feeling the effects of the vaccine. So you tend to get either a slight fever. Some people get pretty strong fevers, right? Or you feel dizzy or nauseous, right? Is because your immune system's working. Okay. Some people don't feel anything at all, which is okay, right? Um, so ultimately, right, from the exposure to this vaccine, you'll develop memory B cells, and these memory B cells will produce more antibodies, right? More antibodies. So I want to kind of emphasize that if you're infected again by the pathogen, okay? If you're infected again, your memory B cells would basically be active already to produce the antibodies, okay? So I want to just show you guys this little representation for you guys to take a look at how it works, right? So this is the virus, right? And um, on this virus, you have particular parts, right, that create an infection, and these parts of the virus, they're basically the active sites, if I can say, right? So don't worry about these fancy words, epitopes and paratopes. Um, that's not what we're focusing on. But when you get an infection and you start producing antibodies, these little circles that you see here are the antigens, and essentially these antigens have little receptor proteins that your antibody will come in and bind to, right? So you have these little antibodies. Whenever they bind to the antigen, that basically nullifies. All right. What does nullify mean? To nullify something means to what? Liu? What does to nullify mean? To nullify or neutralize, right? To neutralize the antigen. So that's the key point that biologists and medical scientists try to go for. That's the end goal there. You want to stop these antigens via antibodies. Okay. So make sure you understood that, right? It's just a simple representation of antigen antibody. So like I mentioned before, vaccines, they consist of dead, weakened, or inactive form of the pathogen. One way or another is going to jumpstart your immune system. Generally, microbiologists, they come in and they remove the DNA and RNA to prevent replication, but the particles of the virus, for example, can still provide that physical structure of the antigen, okay? So again, this is why I'm referring to it can contain an antigen component from an infected organism, okay? So that's essentially the goal of vaccination, right? If a vaccine can stimulate the immune response, but not a disease in your body, then it was successful, right? You've been immunized against future infections. Okay, so just to give you a little kind of perspective of how B cells work, right? So the spleen and bone marrow work together to produce these B cells, right? And let's say you have a pathogen here, right? This little purple pathogen here comes in, it infects a B cell. Unfortunately, that B cell has to sacrifice itself, right? It's referred to as a naive B cell, okay? But at the same time, it is able to register, this B cell registers the antigen for this particular disease. And then it kind of registers and remembers the structure of the antigen, and that is passed down to other B cells. So now you have a whole arsenal of B cells that they keep replicating and they keep dividing and dividing and dividing. So you have a whole library of memory B cells. Okay, so if you were to get this infection again, these memory B cells would be waiting, if I can say, right? And this has been happening since you're born, right? To try to fight off any infection for the second time. That's why generally you don't feel as bad if you get I don't know, if you get that seasonal flu, you get it once and then you really don't feel it the second time, but that doesn't mean it's not in your body, right? It's just that your body has become more efficient to fight it off. So essentially, B cells is this, right? You have this little cell looking at a poster, seeing this wanted guy, this little pathogen, and then he sees him and basically kills him off. Okay? Any questions on that? Good? All right. So remember, we're talking about strategies on how to prevent the spread of pathogens, right? We've talked about quarantining. We've talked about vaccination. Now, herd immunity, right? Herd immunity. What is that? I'm sure this is one of the things we've heard about. We've heard about many times during COVID. But in essence, it's essentially getting the whole community, right? Getting a whole population and trying to vaccinate it, right? To prevent a spread, right? To prevent that propagation of a disease. Okay. Essentially, it's whenever many people in the community are immune to a contagious disease to essentially slow it down or completely stopping it. Right? This is called herd immunity. Now, individually, you don't get affected if you're vaccinated, right? It doesn't really help you in that sense. But for the greater good of the community, it does help slow the spread down, right? And of course, this does depend on the type of virus or the type of pathogen, for example, like measles, right? But it doesn't work for all diseases. Not all diseases are spread or are very contagious. For example, like tetanus, right? You guys remember, okay? I'm gonna zoom out for this, but there's really no way around this particular principle of herd immunity. So I'm gonna highlight some of the key things that you need to know here. But essentially, you do need to know these 7 things, right? These 7 principles, if I can say. Okay. So in terms of principles to start herd immunity, right? You need to have a population high enough in terms of proportion to for this to actually function, right? If you see in this graph here, right, there's only, of course, the orange individuals are those that infected, purple and black, blue and black. Blue are immune naturally. Black are susceptible individuals, right? So the more people you have vaccinated, the more likely it's difficult for the infected individual to reach those susceptible individuals. But it doesn't work if the population is low. So you need to have a high enough proportion of the host population to be immune for it. And this could depend on the disease. Some diseases are more virulent than others. So the jump from one individual, the transmission is a lot faster. Okay. Number two, the immunity and herd immunity is generally done by a vaccine, right? You can't really refer herd immunity to when everyone is naturally getting immune, right? That doesn't really work. It's going to take too long. So the term you implement this term whenever you're referring to a artificial vaccine that has undergone, right? As the case of COVID, right? We all immediately started getting vaccinated. Okay. Again, with that same idea of antibodies and antigens, right? I won't go again into that. And of course, by having the vaccine, you'll start limiting the spread of the disease, right? Or the spread will start to decrease. Okay, for that same matter, like I just mentioned before, more people are immune. It's more difficult for the infected individuals to go and infect susceptible individuals. Okay, so if you notice in a population without herd immunity, there's a lot more um, susceptible individuals. Of course, spread will increase. Okay, so in our model of the lab that we did, all of you were susceptible individuals. Nobody was vaccinated. We could have done that if we, let's say, maybe flip the coin. If you're vaccinated or not, you'll be immune, but that would be a little bit more difficult to model. Okay. So, and then number four is probably that key principle thing that you need to know, what I've been saying here, right? So infected hosts are more likely to come into contact with immunized individuals or individuals that have had the vaccination. Okay. And of course, there's a relationship. The higher vaccination rates there are, or more individuals, the more likely the disease will decrease, or the greater the protection. Okay. There are some limitations, and I'm going to kind of emphasize on that for number six, right? Not everyone can get vaccinated, right? There's some people that are immunocompromised or they have I don't want to say brittle, but fragile bodies that they can't cope with vaccines, or as we've probably seen, have heard, uh, ah, pregnant, no, no, mothers, right, that are basically in their pregnancy, right? They have difficulty. It's a lot more difficult for them to get vaccinated, right? Just by the sensitivity of the baby, right? Uh, yeah, so these additional parameters basically kind of describe what herd immunity is. Okay, like I mentioned before, for number seven, um, this does depend on the virulence of the pathogen and how transmissible it could be and how strong it is. Okay, the higher the virulence, the faster it could be transmitted. Okay, so herd immunity. Any questions on that, guys? So that's another kind of management strategy. So make sure you understand different management strategies there. Good. And these are just some little statistics here in terms of vaccination, how it has helped, right, humanity in terms of survival rates, in terms of infection rates um, throughout the years, right? So you can see here at these points, right, these are the points where the vaccine has been introduced and implemented and if I can say activated, there's a great decrease, right? This is the influenza vaccination program, 1990s, and the amount of infected individuals greatly decreasing, right? Meningitis, right? Basically coming from a meningococcal um, sure, viral disease, right? Early 2000s, right? After that, the infection rate will essentially go down, right? So you do see a trend, a positive trend, that vaccinations can help lower the rate of infection just because you become immune by this method. Okay. And figure 14.6 is just um, a summary, right? A summary diagram for you to understand what I've been talking about for the past 20 minutes. All right, so I won't go over that. Okay. All right. So another factor or another management strategies is disruption of a pathogen life cycle. So this one I've talked about before. It shouldn't be completely new. And it's essentially understanding the life cycle of a pathogen or a vector that at critical points you can stop the life cycle, right? And prevent the spread of disease. Okay. So uh, just to kind of reiterate the pathogen life cycle, if you guys remember, for example, like the mosquito and Plasmodium, you have the human, even within the human, you have muscle tissue having a certain life stage. Then you have liver tissue having a certain life stage, then going to the mosquito and then back to the human, right? Sporozoites, right? Either way, um, you need to remember these, right? The life cycle, exit and entry portals or pathways, the reservoir host, the method of transmission, right? How it actually replicates and reproduces inside the individuals, and the susceptible host. The majority of cases, the susceptible host being humans. Okay, so again, this make sure you remember. I won't go over every detail of that because we've just gone over it couple of sections before. But what you do need to emphasize on is what you want to target, right? If you want to stop this pathogen life cycle, generally you want to disrupt its life cycle. So that's a key word for you guys to use in terms of, um, you know, being more technical, right? Disruption of the pathogen life cycle, and it could be any of these, right? Transmission, the entry point, replication, um, its persistence in the reservoir host or its ability to thrive in the reservoir host, how it exits that host and goes into the other organism, and so forth, right? In terms of replication, right? Um, we know a lot in terms of viruses. We know that there's a lot of restriction enzymes that are very, very specific to viruses. We know that bacteria use binary fission, a reproductive method that is very different from humans, from eukaryotes, right? Um, fungi and protists, they also reproduce um, not just asexually, they undergo a different type of fission. It's called multiple fission, where they can produce instead of two, they can produce three or four or five individuals at once. So all of these kind of factors scientists try to look at to try to stop um, replication, right, and to stop the life cycle. Okay. So I mentioned persistence. Persistence, a relatively new term for you guys, relates to survival in reservoirs. Okay. Or viability outside host. Essentially, how well it could thrive outside the host. Okay. Persistent. How persistent is this pathogen, right? And how difficult is it to stop? Okay. And what defines it? What defines when you stop an organism? Basically, when it can't do the things that I just explained, right? One way or another, one of these factors will stop, right? Um, Transmission, infection, replication, et cetera, right? Ultimately, the end goal being eradication of the disease. Okay. So how do you kind of regulate that, try to control that? These same three factors that we talked about: transmission, susceptibility of the host, and growth of the pathogen, right? Those modes, those three factors that are interrelated, right? If you can stop these, you stop it. You can essentially eradicate the disease. Okay. An example that is used a lot is Plasmodium. Basically, the that uses the mosquito as a definitive host, the human as an intermediate host, right? Ultimately, this that gives you malaria, right? So I've been talking about stopping these different things. So how do you stop? How do you stop it? Well, via pharmaceutical treatment. So if you take a look at this um, diagram here, this is essentially what I've been talking about. All right. If you take a look at this diagram and you can detect that the individual has been diagnosed with this particular disease, you can use medicine, for example, chloroquine, okay, or primaquine. These are two similar types of pharmaceutical drugs that are used to kill the actual plasmodium and the different forms, in this case, sexual forms, right? And if you were to take that, right, if the human takes this medicine, they can kill essentially the plasmodium from even going to the mosquito, right? Not just drugs, not just pharmaceutical drugs, but also how can you prevent the spread, right? Basically, prevent biting, prevent the entry from these mosquitoes one way or another to come in and bite and blood feed on another individual, right? So one way or another, you're preventing the mosquitoes from biting you. Okay, by medicine, physically inhibiting mosquitoes in your daily life. All right. So basically, denying a blood meal protects from mosquito bites. Okay, you just stop immediately the cycle. Okay. Any questions there, guys? And I'll finish off just with these little control strategies, moreover related um, to mosquitoes, mosquito-borne diseases, right? We've always been talking about Plasmodium and mosquitoes there, widely targeted for control in many countries, right? Um, fairly, I don't want to say fairly easy. There's still a pest throughout the world, right? But it's easier to create insecticides for mosquitoes, right? This type of insect that has a biology much different than humans, right? So you can create insect targeted repellents, right? And in this case, you could have some larval targeted drugs or insecticides. Aside from that, like the chloroquine that I mentioned before, that could actually um, um, prevent the plasmodium from developing, right? These are referred to as anti-malarial drugs, okay? So one way or another, you can try to stop the development of mosquitoes or the plasmodium, right? Or the actual pathogen. So um, there's good strategies in this case, in particular with mosquitoes and malaria, but there are some kind of drawbacks, if I can say, right? Um, it is a little bit difficult to um, have early diagnosis, okay? Um, just because of a lack of diagnostic tools in different areas around the world, right? Poverty, healthcare access, right? Education in the sense of telling people to be careful with um, just exposing themselves to mosquitoes. Okay. For the second and third point, resistance, right? You always, this is kind of one of this one of these forever going problems in terms of medical science, that you're always going to have resistance, microevolution occurring. You need to create new drugs, right? So killing asexual and sexual parasitic forms, right? Um, Drugs could follow, but you'll eventually get some resistance, right? Even for the pathogen, for the parasite, and for the mosquito itself, right? You could get some resistance there. So there's always that problem. Okay. Um, of course, poverty, health access, education. One way to prevent, maybe in Japan is not so common. Definitely in tropical countries, especially countries that don't have good higher poverty levels. A lot of people don't have um, um, air conditioner in their houses, so they have to keep the windows open. So what do? They actually have little nets that they put in their bedrooms, on their beds, right? They're like little Cinderella nets. Have you guys seen that? Right? It covers all their bed, so they're protected from mosquitoes, right? Um, Maybe if you've gone backpacking or camping, you would use this, right? But it works, right? Bed nets, closing your windows, repellents, right? One way or another, you want to reduce the transmission. Okay. One way or another, you want to reduce the transmission. Okay, but of course, not everyone does this, so there's a problem. All right, there's no full way to guarantee that everyone's on board with that, right? And then lastly, right, humans that are traveling around, um, preventive medication, right? Trying to get preventive treatment to avoid spread. Also, not necessarily for malaria, but for other diseases, you get vaccinated, right? Um, uh, you want to one way or another prevent, right, via vaccination. That's why, if you've probably heard, before you travel, it's always good to get like booster shots of vaccines, right, before you travel. Okay. Now, lastly, sorry, sequencing, um, and more specifically with viruses, right? Sequencing, you guys remember, is basically understanding the DNA sequence. If you know the DNA sequence, you know the gene, you know the protein, right? So by understanding better sequencing of DNA, you can create better vaccines. Remember, antigens, just like antibodies, are proteins. So if you know the specific protein of the antigen, you can create more higher fidelity antibodies. Okay, so sequencing has been a big breakthrough in vaccine development. Okay, some examples are block sporozytes, different developmental stages of the plasmodium, and not just blocking, but you can actually destroy them, right, with sequencing techniques. Okay. This map that you see here is a kind of predicted modeling based on current data, right? Um, what is the likelihood of certain diseases to be spread? In this case, you have the Plasmodium malaria disease, how it's spreading out throughout the world based on our current conditions, right? You see, it's interesting. It's actually going upper to more temperate regions, cooler regions, but you see that it's more limited to that warmer equatorial regions there. Okay, climate change, guys, it's real. All right, guys. Um, so that's good. I think that's it for today. We have some good timing. So if you guys notice, there's essentially here 4 to 11. Don't freak out on all these different pages here. That is not everything. So the last page of actual content is actually these 4 pages. So, and we'll be done with the entire WACE program. All right. What I do want to show you guys from page 11, 12, 13, and on are just tables for you to summarize. Okay. So start studying that and reviewing, right, for all the content. Take a look. Oh, sorry, and this last portion. 14.3. Good.